Welcome to our next class, that is Kingdom Monera. So today we are going to learn about the Kingdom Monera. So normally in this particular kingdom we are going to study is the first Kingdom Monera, and this particular kingdom contains prokaryotic cells. So the Kingdom Monera includes the most ancient, the primitive form of organism with a very small and very simple. And so these organisms are called as microorganisms. So Monera includes the most primitive and simplest form of organism. Mostly they are prokaryotics as they are primitive forms, and these organisms are regarded as the first inhabitants of the earth. So these organisms on the evolution point of view regarded as the first inhabitants of the earth. And bacteria is regarded as the sole member. Bacteria is regarded as sole member of the Kingdom Monera. This is a very important point. The Monera contains bacteria. So we are going to discuss about bacteria in this particular chapter. So let's talk about bacteria. So we are just going to discuss a brief account of bacteria and the detailed cell structure, its life process detail we'll study in the prokaryotic cell, that is in the cell chapter. Okay, bacteria is almost present everywhere. Means they are cosmopolitan. They are cosmopolitan in their habitat, means found everywhere in the world, and hundreds of bacteria are present in the handful of soil. It is when we go for any handful of soil, so present hundreds and millions of bacteria present in a handful of soils. So bacteria can live in extreme habitats. Bacteria can live in extreme habitat or harsh habitats, like they are found at a hot spring, they are found in deserts, they are found in snow, they are found in deep oceans also. Many bacteria lives as parasite on other organisms. So they live as parasite. So as the bacteria parasite, so they are what? Disease causing. They are pathogenic in their nature. And most Monerans are primitive organisms, so they can flourish in different organism environment. Normally, move to the characteristics of Monera, we find these are either unicellular organisms or they are present in colonies, they are present in groups in colonies, or they are present in filamentous form. Either unicellular form or present in groups called as colony, or they are present in filamentous forms. So this is the arrangement of habitats of bacteria. Now they are mostly prokaryotic organisms, as they don't contain nuclear membrane, they don't have membrane bound organelles. So these are prokaryotic organisms. As we know, the nuclear membrane, the nucleolus, and the histone proteins are absent, which are the condition of eukaryotic organisms. These nucleate, the naked DNA, this bacteria contains nucleate, and this nucleate is a naked DNA present, means they don't have nuclear membrane. As they don't have nuclear membrane, the DNA is exposed in the cytoplasm, so it is called as the naked DNA, and it is mostly circular DNA, and also it is double stranded. It is mostly circular DNA and it is mostly double stranded DNA. Got it? They contains RNA. Yes, they contains three basic types of RNA. That they contains mRNA. They have mRNA, they have 70S, that is rRNA, they contains rRNA, and also they contains tRNA. And the RNA present mostly are inside the ribosome, rRNA, that is ribosomal RNA. This ribosomal RNA is present inside 70S ribosome. Inside 70S ribosome. What is 70S? We'll study details in the cell structure. Got it? So now let's move. And the DNA is circular. Yes, the DNA is circular. And it is double stranded. DNA is circular and double stranded. But when we talk about the bacteria, archaebacteria, mycoplasma don't have no cell wall. They don't have cell wall. But archaebacteria contains cell wall with a modified form so that they can survive in harsh and primitive conditions. But mostly bacteria contains in the cell wall a chemical that is called as peptins present and glucose present. So peptidoglycan is present. It is sometimes called as murine. We'll study detail in the cell chapter. It is sometimes called as murine. Whereas archaebacteria don't have murine, they have branched lipid chains. We'll discuss later in our cell chapter. Okay. Now let's move to the structure. We find the outermost structure. The outermost structure normally present is, that is the capsule. So capsule is the outermost structure. Next to the capsule we find the cell wall present. Next to the cell wall we find the cell membrane present. So these are the outer boundary or the outer covering. These are the outer boundary. Inside the boundary, we find the fluid present called as cytoplasm. Inside cytoplasm, we find 70S ribosomes are present. This ribosome present, it is a 70S ribosome. 70S ribosomes are present. And we find a naked DNA present, that is called as nucleoid. And this nucleoid contains naked DNA. This nucleoid contains, that is called as naked DNA. And also we find some inclusions are present, like gas vacuoles present, volutin presence, phosphate granules are present, etc., etc. And also we contains, in some bacteria, not all bacteria, we contains extra chromosomal DNA present, that is called as plasmid. This is called as extra chromosomal DNA. Extra chromosomal DNA. So we find extra chromosomal DNA. And in the outer part, we find certain appendages present. A long appendage is present for movement, that is called as flagella, which is made of flagellin protein. We find small, then we find attachment to attach with any organism, which is Vibrio present. And also we contain spilia that do help in reproduction. That is sex spilia present that do help in reproduction. So this is the brief structure of the bacteria, that is the bacteria present in Moneral kingdom. But we'll study the detailed structure, what is, how cell wall is made up of, water capsules made of slime, loose slime, all we'll study in the chapter that is called as the cell chapter. Okay. Now, let's move. So we find that in bacteria we don't find membrane bound organelles are present, like mitochondria, endoplasmic reticulum, Golgi body, etc., etc. They all are absent. And the ribosome present is of 70S types. And in some bacteria, they are photosynthetic bacteria, some are non-photosynthetic. So these are photosynthetic bacteria, special bacteria present. These are photosynthetic bacteria which prepare their own food. Then the respiratory enzymes are also present in the cell membrane. Suppose this is the cell wall is present. For example, this is the cell wall present. So inside the cell wall, we find enfoldings are present. These type of enfoldings are present. And these enfoldings are called as mesosomes. These enfoldings are called as mesosomes. Inside mesosomes, we find the respiratory enzymes are present. So inside mesosomes, we find the respiratory enzymes are found to be present. Got it? So inside mesosomes, so these are the enfoldings of the plasma membrane called as mesosomes. Mesosomes. And the mesosomes is of two types. One is the septal mesosome, another one is called lateral peripheral mesosomes. And these mesosomes contains the respiratory enzymes for respiration. So respiration. So normally the respiratory enzymes helps in performing, that is called as the Krebs cycle. So the mesosomes are acting as the primitive mitochondria. They are the mitochondria, primitive mitochondria that helps Krebs cycles to produce ATP. Got it? But in the cytoplasm we find that is glycolysis takes place. So glycolysis takes place in the cytoplasm. So normally the release of ATP or the release of energy currency is from a special structure plasma membrane that is called as mesosomes, which behaves as mitochondria because it do contains the respiratory enzymes to maintain the Krebs cycle, the process that do helps in producing ATP. Okay. Now, reproduction is mainly asexual. Mainly it is asexual mode because they don't have gametes. So the reproduction is without gametes. It is without gametes. Got it? And bacteria show both autotrophic nutrition and heterotrophic nutrition, as we have discussed earlier. So bacteria show both autotrophic and heterotrophic mode of nutrition. Autotrophic bacteria synthesize food from inorganic substances. They produce organic compounds. They produce organic compounds. From inorganic substances, they produce organic compounds. Got it? And photosynthetic autotrophic bacteria, they need light energy. And for chemosynthetic bacteria, they need chemical energy. So photosynthetic and chemosynthetic both are autotrophic. Both are what? Now autotrophic organisms. So heterotrophic bacteria often ready-made food. So they depends on autotrophs for their foods. And now heterotrophic bacteria are pathogenic. They are normally, some are pathogenic. Some are friendly and some are pathogenic. Heterotrophic reproduction is mostly that is saprophytic, means they depend on dead organism. So they extract the food from the dead organic matter. So mostly they are what? That is saprophytic. Some undergo symbiotic mutual benefit and some depends on living host. So parasite, they depends on living host. And saprophytes are dead, so they are decomposers. So these bacteria are decomposers and parasite is a pathogenic. These are pathogenic. And symbiotic, they are mutual benefit. The health and mutual benefit. We find mutual benefits bacteria present. Got it clear? Okay, next let's move. Shape of bacteria. So the bacteria classified on the basic shape. Coccus, a spherical shape, means the bacteria are spherical in nature. So these are called as Coccus, a coccid. Bacillus, if the bacteria is a rod shape like structure. So this is spherical structure and this is a rod shape like structure. So Bacillus. Vibrio, if the bacteria is comma shape like structure. If the bacteria is comma shape like structure, it is called as Vibrio bacteria. Spirillum, is the bacteria is spiral in structure. If the bacteria is spiral in structure, they are called as Spirillum bacteria. So the plural form of Coccus, Cocci. Got it? Bacilli, got it. So these are the basic shape. So this is the Coccus form, this is the Bacillus form, and this is the Spirillum form, and this is the Vibrio form. But we have different shape of bacteria. There are different types of bacteria present, and the Cocci is again classified into different types. The Bacillus is also classified into different types, and other bacteria is also classified. Let's discuss one by one about the classification of bacterial shape. Okay, let's discuss about the different shape. So let's talk about the first shape. So when we move on to the first shape of bacteria, let's discuss about the Coccus form. This Coccus is again classified. It is called as monococcus. Monococcus. So monococcus bacteria, if it is free living, only single bacteria present. Diplococcus. Diplococcus. So Diplococcus we find if the bacteria are present in colonial form and it's 2 in number. Streptococcus. Streptococcus. In Streptococcus we find that group of colonies present in chain form. Streptococcus we find they are present in chain form. Staphylococcus. Staphylococcus. In Staphylococcus we find they are present in bunch, grapes like form. If they are present in bunch, they are called as Staphylococcus. Sometimes also we find that the bacteria are present in 8 in number, 4 in number. If they are present in 8 in number, and these bacteria are sometimes called as Sarcina. These bacteria is called as Sarcina. And sometimes the bacteria are present in 4 in number, in 4 colonial form. It is called as Tetracoccus. Tetracoccus. So these are different forms of Coccus bacteria. So let's go for this example. In monococcus, which group of bacteria is regarded as monococcus? That is Micrococcus luteus. This is Micrococcus luteus is the example. And Diplococcus. Diplococcus is Diplococcus pneumoniae. Diplococcus pneumoniae, that causes the pneumonia disease. Then move on to that is called a Streptococcus. In Streptococcus, the best example that is called as the present Streptococcus lactis. Streptococcus that is called as lactis. And Staphylococcus which are present in bunch, grapes like structure. And the Staphylococcus example is called as Staphylococcus aureus. Staphylococcus aureus. And when we move on to Sarcinia, that is called Sarcinia lutea. Sarcinia lutea is its example. Sarcinia lutea. And when we move on to Tetracoccus, the best example is called as Neisseria. So these are the examples. So monococcus, we find free living bacteria simple. That is the example Micrococcus luteus. We find Diplococcus present in 2 in colonial form. Diplococcus pneumoniae. We find Streptococcus which is present in chain like forms. And we find Staphylococcus which are present in bunch. That is called as Streptococcus lactis is for Streptococcus, and Staphylococcus aureus is for bunch. And we find present in 4 in number, that is called as Tetracoccus, and that is called Neisseria. And we find sometimes Sarcinia which are present 8 in number in 3D structure in a colonial form, and this is called as Sarcinia lutea. So these are the different shapes of bacteria of Coccus. Now let's discuss about the different types found to be present in the bacteria that is called as Diplococcus. So when we move on to Diplococcus, let's talk about let's talk about that is called as Bacillus. That is rod shape. Now let's discuss about the different forms present in the rod shape bacteria. So rod shape bacteria is simple if they are present in free form. If they are present in simple free form, that is called as monobacillus. Monobacillus means these are present in simple form, and the best example of monobacillus is called as lactobacillus. Lactobacillus is the best example of monobacillus. And the second one, that is called as diplobacillus. Diplobacillus. So diplobacillus, they are present in colony of 2. Diplobacillus, they are present in colony of 2. And the best example of diplobacillus is called Bacillus subtilis. Bacillus, that is called subtilis. Bacillus subtilis. And the third one, that is called as streptobacillus, which are present in the chain. Streptobacillus. They are present in chain, colonial chain. So if they are present in chain form, that is called as streptobacillus. And the best example is called as streptomyces. Streptomyces. There are certain bacteria which are present in, that is, stalk-like form. There are certain bacteria which are present in stalk-like form, for that is called as palisade. Palisade bacillus. Palisade bacillus, which are present in, that is, stalk-like form. Palisade bacteria. Got it? So these are the different shapes of bacteria found to be present in bacillus. This is called as monobacillus, free-living bacteria. Example is lactobacillus. Diplobacillus, 2 in number, colonial form. Bacillus subtilis. Streptobacillus, they are present in chain, and palisade bacillus, they are present in stacks. So these are different forms of shapes present to be present in the form that is called as bacillus. Now, some bacteria are present where we find that they are stalked bacteria. Some are stalked bacteria. Stalked rod-shaped bacteria means these bacteria are rod-shaped, but they have stalk-like structures. So as the stalk, the best example, that is called as Caulobacter. The best example is called as Caulobacter. And some bacteria, they are present, they are pleomorphic. Pleomorphic means they change their shape. They change their shape accordingly. And the best example is called as rhizobium. Rhizobium is the best example. And this pleomorph, sometimes it becomes Coccus, sometimes it becomes Bacillus, according to the nature of the surrounding. So any organism, any bacteria that change its shape according to the change of the surrounding is called as pleomorphic, and the best example is that is called as rhizobium. And later in the chapter cell, we'll study about the gram-positive bacteria. There are certain bacteria present that is called as gram-positive bacteria on the basis of cell wall, and that is called as gram-negative bacteria. So we'll study later this in the chapter that is called as cell. So this is all about the general character and general shape of the bacteria. In the detailed structure of the bacterial cell and its functions, we'll study in the chapter that is called as prokaryotic and eukaryotic cell. Okay, now let's start. That is bacterial life process. So normally this bacterial life process is involved in 2 parts. One is respiration, other one is called as nutrition. So we'll learn detail about the respiration and its nutrition. So what is respiration? The respiration is, that is, oxidation of food materials to release energy, or metabolic activities called as respirations. And the respiration is again of 2 types, either in presence of oxygen or in absence of oxygen. Absence of oxygen. Got it? So either aerobic respiration or anaerobic respiration. So what is respiration? Respiration is called as oxidation of food to release metabolic energy, or various metabolic activities. So metabolic energy in the form of ATP. So respiration either in presence of oxygen, because oxidation, or in absence of oxygen. So this is called as aerobic and anaerobic respiration. Okay, and bacteria are divided on this basis of aerobic and anaerobic into 2 different forms. What are they? One is obligate, another one is called as facultative. So what is obligate? Obligate means that throughout the lifespan, the bacteria only use oxygen to survive. Means they are aerobic throughout the lifespan. They are aerobic throughout their lifespan. So this is called as what? This is called as obligate. So obligate means presence, they use oxygen. Aerobes means, strict aerobes means they use oxygen throughout the lifespan. Means they always survive in aerobic condition. If anaerobic condition comes, then they die, unable to survive. Then they possess enzymes for aerobic respiration. Yes, to maintain aerobic respiration, what all enzymes are needed? They have all the enzymes present. And the best example is called as Bacillus subtilis. So Bacillus subtilis is the main example of obligate, or that is called as strict aerobes. Then move on, what is facultative anaerobes? So normally they are aerobic. Yes, facultatives are normally aerobic, but in adverse condition. They change to anaerobes. They change to so called as facultative anaerobes. In adverse condition, they switch to anaerobic condition, and this nature is called as facultative. The best example is called as Pseudomonas. So facultative are normally aerobic. They are normally aerobic, but in adverse condition they change to anaerobic, and this nature is called as what? Facultative. Got it? Then move on to obligate anaerobes. Since obligate means what? Strict means throughout the lifespan they behave as anaerobic. So bacteria respire anaerobically throughout its lifespan. So throughout its lifespan the bacteria respire anaerobically. And in this condition the growth is very slow because less energy is utilized. Less energy means 2 ATP will be released from anaerobic respiration per glucose molecules. But in aerobic condition we release 38 ATP per molecules. But here as the energy is less, so the growth is also very less. And the best example is called as Clostridium botulinum. So Clostridium botulinum is the best example of obligate anaerobes. Facultative means normally they are anaerobes, but in adverse condition they change to, that is, aerobes. So normally they respire anaerobically, but in adverse condition they change the mode to aerobically, and this is called as facultative. So facultative are graded, they are highly pathogenic because they change according to the change of the surrounding. And the best example is that is called as photosynthetic bacterium, that is chlorobium. So chlorobium is the best example of facultative aerobes. What is facultative? Normally they are what? Normally they are anaerobes, but in adverse condition they change to aerobic mode. Now let's classify the nutrition. So what is nutrition? That is synthesis of its food materials or extracting its food materials. So bacteria classified on the basis of nutrition into 2 types, and it depends on the carbon source. And the classification of bacteria always depends upon the carbon source, because to produce organic or inorganic compounds they need carbon materials, and it also depends on energy and electron transport. So not only carbon, they also need energy, and also they need electrons to classify the nutrition. So the first one, that is called as the first part of nutrition, that is called as photolithotropic. So photo, light, in presence of light. Lithotropic means from inorganic substances to produce organic substances, and it is of 2 types. One is called as purple sulfur bacteria. They use the pigment that is called as bacterioporphyrin pigment, and they use inorganic sulfur, inorganic compounds, and the donor, the electron donor and H plus donor are present from this sulfur compound. The best example is called as chromatium and thiospirillum. So on the basis of photolithotropic bacteria, one is purple sulfur bacteria, another one is called as green sulfur bacteria. Purple sulfur bacteria use bacterioporphyrin, whereas green sulfur bacteria use bacterioviridin. So bacterioviridin pigment or chlorobium pigment is used by this green sulfur bacteria. And H2S, that is E minus H plus donor, is extracted from H2S, and the best example is chlorobium. So photolithotropic bacteria uses light as its energy. So they use light energy. So the solar energy is used by this bacteria. So it is called as photo. And they synthesize complex food from inorganic materials with the help of the pigment that is called as bacteriochlorophyll or bacterioviridin. So they use light, they use inorganic compounds, they use inorganic compounds. So they use inorganic compounds, that is lithotropes, litho. And it is of 2 types. One is called as purple sulfur bacteria, another one is called as green sulfur bacteria. The purple sulfur bacteria contains bacteriochlorophyll as the pigment to produce synthesized food, and the green sulfur bacteria contains bacterioviridin. The green sulfur bacteria contains bacterioviridin and the purple sulfur bacteria contains bacteriochlorophyll. And these are photosynthetic bacteria, but they never release oxygen. They never release oxygen. So they are non-oxygenic. Non-oxygenic photosynthesis. Or it is called as anoxygenic photosynthetic bacteria. Because hydrogen acts the reducing power. Electron is released from this hydrogen. Got it? And no oxygen is evolved during this process. So no oxygen is evolved. So water splitting is also not done. That means water is not broken down to oxygen or H plus ions. So no release of oxygen. And the reducing nature comes from, that is H2S or from the sulfur. That is directly it comes from sulfur or from H2S. So like carbon dioxide is the raw material used and they use H2S. They use H2S. Solar energy is used. So sugar is produced plus sulfur is produced. Not oxygen, no oxygen. Sulfur is produced and water is produced. So this is an oxygenic photosynthesis. Got it? Photoorganotrophic means they use organic compound to produce organic materials. These bacteria use organic matter, so it is called as photoorganotrophic. But the bacteria that uses inorganic matter is called as lithotrophs. And organic matter acts as electron donor. And in the last we studied litho, there inorganic compounds acts as electron donor, but here organic compounds acts as electron donor. Got it? And here organic matter also acts as carbon source. And this photoorganotrophic bacteria that, as we have already discussed, it is called as purple non-sulfur bacteria because it is organic. So it is non-sulfur bacteria, like the green non-sulfur bacteria. It is organic. So the best pigment is bacterioporphyrin for purple non-sulfur and bacterioviridin for green non-sulfur. And they use the aliphatic organic compounds. They use aliphatic organic compounds as electron and hydrogen donor. The best example is called as Rhodospirillum and Rhodopseudomonas for purple non-sulfur bacteria. And for green non-sulfur bacteria, it is Chloronema. The best example is Chloronema and Chloroflexus. Got it? So this is all about photoorganoheterotrophic. So we got two types of bacteria present. One is that is photolithotropic. One is photolithotropic bacteria. Another is photoorganotropic. In lithotropic, inorganic compounds are used. In here, what is organic compounds are used. So this is all about the nutrition of this particular bacteria. So purple non-sulfur bacteria. Let's discuss about purple non-sulfur bacteria. So they contains bacteriochlorophyll as the pigment to produce food materials. Organic compounds are used as electron donor. And the best example is Rhodospirillum and Rhodopseudomonas. Green non-sulfur bacteria. They contains bacterioviridin as the pigment and non-sulfur organic compounds acts as the electron donor. And the best example is Chloronema and Chloroflexus. Got it? This is all about your green non-sulfur bacteria. Now let's move to the next part of autotrophic, that is called chemosynthetic. So we have discussed the first one, that is called as phototrophic bacteria, which is of two types. One is photolithotropic, another one was photoorganotropic. And now we are going to discuss about chemotropic. They use chemicals. They use chemicals to produce food, and later last time we used photolithotropic, they use light. So here these bacteria synthesize food by oxidation. Yes, it is by oxidation process. And the energy is obtained from inorganic substances. So here the energy is obtained from inorganic substances for oxidation process. And light is not the source of energy. Here light is not the source of energy, but chemical are the source of energy. So ATP molecules trap the released energy. So ATP molecules trap the released energy and they recycle the nutrients into nitrogen and sulfur. So they recycle into nitrogen and sulfur. So we call them that nitrogen chemosynthetic bacteria or sulfur chemosynthetic bacteria. So they help in recycling iron, they help in recycling sulfur. Let's take an example, that is hydrogen bacteria. They helps, hydrogen chemical is used to synthesize food, and the example is called as Hydrogenomonas. Nitrifying bacteria, Nitrosomonas and Nitrobacter. Sulfur bacteria, Thiobacillus and Thiooxidans. So these are the iron bacteria, that is Ferrobacillus. And iron bacteria is also called as Leptothrix. So these are the certain examples of chemosynthetic bacteria, and these bacteria obtain energy from oxidation reaction from the inorganic compounds. Chemoorganotropic heterotrophic bacteria. These bacteria synthesize food from raw materials. So chemoorganotropic, these chemoorganotropic heterotrophic bacteria are the bacteria that synthesize the food from the raw materials. and they absorb nutrients from the dead matter or from the living host. So this includes organic matter. So dead matter, it may be saprophytic or saprobiotic, or it is parasitic, or it is symbiotic, either parasitic or symbiotic. Yes, accordingly we know what it may be: saprophytic for dead, symbiotic for mutual benefit, parasitic it extract from living organic matter. Got it? This is called as chemorganotrophic heterotrophic bacteria. Saprophytic bacteria. These bacteria depends on dead organic matter and they helps in decomposer. So they are called as decomposers. They are called as decomposers. They act as decomposers. Yes, they act as decomposers. And they convert complex organic substances into soluble compounds. And these soluble compounds are again diffused into the bacteria as nutrients. Symbiotic bacteria, they undergo mutual benefit with the host, like E. coli present in inside intestine. Rhizobium bacteria present leguminous plant for nitrogen fixation. They help in fixing nitrogen, and in return they get the shelter and food from the symbiotic host. And this symbiotic bacteria, example, let's take Azotobacter or Bejernikia. They are aerobic nitrogen fixer. Clostridium, which is anaerobic nitrogen fixer. These are the best example. Azotobacter is aerobic nitrogen. Clostridium pastorianum is the anaerobic nitrogen fixation. It helps in anaerobic nitrogen. Lichen is the combination of fungi and algae. It is not included in bacteria, but just told it is one kind of symbiotic association. Parasitic, that depends on living host. The organism extract food materials from living host. So they are what? Disease causing, or they are pathogenic in nature. Matlab, they are disease causing. Host may be plants or it may be animals. And disease causing bacteria are pathogenic. Yes. So parasitic bacteria are mostly pathogenic bacteria because they help in causing diseases. In the next part, we'll study about the part that is called as reproduction. Now let's discuss about the reproduction in bacteria. In bacteria, the reproduction is mostly by means of asexual type. So asexual method is the type of reproduction where there is no use of gametes, means no use of gametes. And this, in bacteria we find sexual reproduction present, but it is a sexual recombination, that is recombination of genes present. No gametes are involved. No gametes are involved. They don't find any gametes present. And the sexual reproduction is not the true sexual mode of reproduction. So true sexual reproduction is mostly absent. Let's start with asexual reproduction first. So asexual reproduction is by means of spores called as sporangiospores. For example, that the bacteria will produce spore, and this is called as sporangia. The sac-like structure is called as sporangia, and inside sporangia the spores are produced, and this spores is called as what? Sporangiospore. Under favorable conditions, the sporangia burst and the spores are released out to give rise to a new individual. The next type of asexual reproduction is by means of oidia or conidia. In conidia, same thing. The conidia produce in a chain like manna, and these individuals are called as conidia. And also oidia, also we find this type structure oidia present, and oidia mostly present in sugary medium. When the condition is highly sugar is present, ample organic matter is present, oidia reproduction do takes place. And conidia reproduction also takes place in certain favorable conditions. Got it? Okay. And under unfavorable condition, adverse condition, endosperms are produced. Means here what happened, the spores are produced outside the body. Here the spores are produced outside the body. So sporangiospore, conidia and oidia, they all are exospore. They all are exospore. But in endospore, we find the spores are produced within the body. Inside the bacterial cell, the spores are produced. So it is called as endospore. Move on to the first mode of reproduction, that is by means of cell division, that is called as binary fission, and it is the common method of reproduction. And binary fission is a type of reproduction which takes place by means of cell division. So here what happens, single cell, they divides to give two daughter cells. And this type of cell division is called as amitosis. Amitosis means no spindle fiber is involved. No spindle fibers are found because it is naked DNA. And also it is sometimes called as direct cell division. It is called as direct cell division. Got it? So by this, two daughter cells are produced, and each daughter cell then develops into parent cell, matures into parent cell, and also they start dividing. to produce many daughter cells like this. And as a result, multiplication, many number is produced. So by binary fission, bacteria multiply itself. This is the most common method. So one bacterial cells divide into two daughter cells as we have discussed, and spindle fiber is normally absent as we have discussed. And also during binary fission, what happened? Multiplication of large number of bacteria takes place in large population. So many bacteria are produced within a few seconds of time. The few seconds of time many bacteria produce, because mostly in E. coli 20 minutes taken for bacteria to divide. So in 20 minutes we'll find two bacteria daughter cells. In the next 20 minutes we'll find many bacteria goes on dividing. So like this, within a few very short period of time, rapid multiplication of bacteria takes place. Growth, when the population increases, when the number of bacterial cells increases, then the drawbacks. What is the drawback? The growth stops. The growth slows down. Why? The shortage of space. Not only shortage of space, lack of nutrients. Nutrients are insufficient for the growth. Not only insufficient nutrients, accumulation of waste materials also prevent the growth. And also certain bacteriophages directly destroy bacteria. Matlab means what? Now what is bacteriophage? A virus infecting bacteria. Virus infecting bacteria. So virus infecting bacteria is called as bacteriophage. So bacteria are destroyed. So these are the drawbacks in multiplication and growth of the bacteria after binary fission. So binary fission normally occurs under favorable condition, but we find certain drawbacks are present. Now let's move into steps of the binary fission. Replication of DNA first takes place. As we know that bacteria have DNA present, naked DNA present. And during the first step, the DNA replicates. The DNA duplicates into two daughter DNA. Then the mesosomes are produced. So what happened? Now we have one mesosomes initially. But at the time of after division of duplication of DNA, the cell membrane produce two mesosomes. So two mesosomes takes place by membrane synthesis. So by membrane synthesis, two mesosomes are produced, and each mesosome will attach with the respective DNA, its respective DNA. Clear, got it? And after that, what happened? The wall formation takes place, and a cross wall takes place to form two daughter cells. So let's move. So this is the plasma membrane, this is the DNA present. So the DNA duplicates into two. The DNA duplicates into two, and here single mesosomes are present, and here the mesosome duplicates into two, and each mesosome now joins with its respective DNA, and each mesosome now separates the respective DNA to the respective side or respective pole. So both the DNA move towards its respective side, and then a groove takes place, and cross-wall deposition of chemicals, minerals takes place, and the wall is forming between them, and it is being separated, and we find two daughter cells are produced. So these are the steps of binary fission. So DNA duplication, DNA replication takes place, as we all know that the DNA of bacteria is the circular DNA and is double stranded. So when DNA replication takes place, first it converts to single stranded form, simultaneously convert to single stranded form. So this is single stranded strand, this is single stranded. So now the DNA will duplicate, new daughter DNA will produce. So this method looks like the theta appearance of Greek. So this model of DNA replication is called as theta model of DNA replication. So replicating chromosome appears like a Greek theta model. Yes, so it is called as theta model. This model is called as the theta model of DNA replication. Mesosome duplication takes place, as we have discussed. Let's suppose this is the parent mesosome present, and this is the cell membrane. So here the cell membrane synthesizes to produce two mesosomes. So two mesosomes are produced by the division of the cell membrane. So mesosome divides by membrane synthesis. Yes. And chromosome move to the daughter cells, as we have discussed. And after that, cross-wall formation takes place, as we all know. From the periphery, the deposition chemicals take place. Already the chromosome and the mesosomes are separated, and this nature is called as the plasma membrane. The cross-wall starts. Not only cross-wall, deposition of chemical takes place to produce cell wall. And initially first they produce cell membrane. So first cell membrane produced, then deposit of wall chemicals takes place to form a cross-wall synthesis that will separate the two daughter cells into individual daughter cells. So new wall materials are deposited on it. Septum and cross-wall synthesis occur. So as a result, two daughter cells are produced. Now let's move into the next asexual mode reproduction, that is called as endospore. endosperms are formed in bacteria like Bacillus. In Bacillus bacteria, Rhodsia bacteria, the endosperms are produced within the bacteria. So these are called as endospore. And the endospore is produced during unfavorable conditions, either at the center or at the terminal. And it is produced within the bacteria, inside the bacteria, so they are named as endospore. So endosperms survive in adverse conditions. Yes, they can survive in adverse conditions. They can withstand highly adverse conditions. So they contain resistance bodies. These are sometimes called as resistance bodies that can withstand adverse conditions. Endosperm may be spherical, may be oval shape, may be terminal, may be central. And they contains a special chemical that is called as calcium dipicolinic acid that provide resistance to the endosperm. And this calcium dipicolinic acid at the time of unfavorable conditions, they help in water absorption. So water absorption takes place at the time of unfavorable condition. Let's discuss. At the center we find the nuclear materials are present and proteins and DNA, RNA all are present. Then we find what? Cytoplasm present, DNA, RNA present, yes. After that we find what? A wall is present. So here we find the DNA, RNA present, then we go for a spore wall. Membrane is present, then spore wall is present. Let's move. So here what happened? The DNA, RNA present, the proteins are present. We find a core wall, the membrane is present, inside the cell membrane is present. Then we find a core wall is present. Then cortex is present. This is the cortex layer. After cortex, the spore coat is present. This is the spore coat present. And then exosporium is present outside. So this is the structure of the endosperm. And when the endosperm matures and favorable condition arises, what happens? That the endosperm ruptures. The endosperm ruptures. The endosperm ruptures and gives rise to the new bacteria. Release the new bacteria. So we came to know that the cortex present. The cortex is made up of peptidoglycan and calcium dipicolinic acid. Very important. The cortex part is made up of calcium dipicolinic acid and peptidoglycan. And spore coat is surrounded by the cortex. First nucleus present, cell membrane present, spore coat present, then we find the cortex present. Then endosperm is highly resistant to desiccation, to chemicals, to unfavorable conditions. That it accumulates protoplast at the center. Protoplast separates the rest of the cells, means the protoplast comes and lies at the center. Here we don't find any protoplast present. And after that, we find the formation of endosperm. For example, suppose the vegetative cell is present. So endosperm start formation either from the terminal or from the center. So accumulation takes place from the center or from the terminal. And when the spore starts formation, it forms spore coat. And after spore formation, the endosperm, then they breaks, this spore coat is broken and released, the new bacterium comes out. So this is the endosperm produced, and the endosperm germinates by breaking the spore coat and giving rise to the new individuals. This is how endosperm germination takes place. So endosperm can resist high temperature. Above 100 degrees centigrade it can tolerate above 100 degrees centigrade. And endosperm is unharmed during pasteurization. What is pasteurization? Heating any liquid at 60 degrees centigrade for 30 minutes is called as pasteurization. And here endosperm can withstand this pasteurization technique. Also, the wall is very thick. They are impermeable to water. The spore coat is impermeable to water, is very thick. So they helps in preventing from mechanical pressure. And low water content increases the resistance, and low metabolic activity also increases its survival. Yes, they have less water content and they have less metabolic activity, so they undergo a resting stage. If they undergo a dormant stage, so in the dormant stage the survival activity increases because the metabolic activity becomes very low. And after that, when the endosperm starts going for germination, this calcium dipicolinic complex present on the cortex wall, they starts absorbing water under favorable condition. So at the time of germination, that starts absorbing water with the chemical present that is called as calcium dipicolinic acid present in the cortex. This endosperm then becomes metabolically active and they breaks the dormant stage, and the thick spore coat ruptures, giving rise to the new endosperm. So germination of endosperm, that means cell, new endosperm rises, a new bacterium arises from the endosperm. So one endosperm will produce one bacteria. So one endosperm regarded as a one parent cell. So one endosperm will give rise to single bacteria. Then in the next class, the next section, we'll study about the sexual reproduction. Okay, let's start sexual reproduction. So yes, sexual recombination, it is not called as sexual reproduction, it is sexual reproduction because no gametes are involved. So bacteria sexual reproduction is primitive. Why? Now no gametes are involved. No gametes used. What happens? A gene recombination, a gene transfer or recombination takes place. So gene transfer or recombination takes place. So this type is called as sexual recombination. It is the primitive form of sexual reproduction. So let's discuss. So in bacteria, the gamete formation of fusions are absent, but there is a fusion of genes takes place, and genetic recombination occurs through this DNA exchange or by DNA recombinations. So methods of DNA recombination, there are three basic methods present. One is called as transformations, is one method of gene recombination. One is called as conjugation, another one is called as transduction. So three methods are involved in sexual recombination. So together we can call them as what? Is the genetic recombination, where the genes are involved in this transfer of characters from generation to generation. Let's move to methods of genetic recombination. So three methods, for example, transformation. So when we move into transformation, in transformation what happens? Suppose this is the donor cell and this is the recipient cells. The donor cells will release its DNA content to the surroundings, and the recipient cells will absorb the DNA components. And these DNA components is integrated into the recipient cells and convert the recipient cells into a donor form cells. This is one type of genetic recombination. Another genetic form of recombination is where the donor cells transfer the DNA or the genetic materials into the recipient cells with the help of a tube that is called as conjugation tube. This conjugation tube, and by this recipient cell converts into a donor forms only by gene recombination. Another one is transduction, where the bacterial DNA is incorporated into the bacterial DNA, and bacterial DNA gets accumulated and they forms a new combination of genes, new combination of characters, and the new bacteriophage so formed contains bacterial DNA and viral DNA, and they again go and transfer the DNA to the other, and this method is called as transduction method. So let's discuss one by one. Transformation. So Griffith, scientists discovered the process of transformation in the year 1928. He experimented on two different bacteria, Diplococcus and Streptococcus pneumoniae, that causes the disease called as pneumonia. And here he used the donor and recipient cells. And the donor and recipient cells never come in contact to each other, but the recipient cell is converted into a donor form. How? Let's move. So donor release the DNA fragment. For example, these are the DNA fragments present in the donor. The donor release the DNA fragments, and the recipient cells, these are donor cells. The recipient cells absorbs the DNA fragments. They absorb the DNA fragments. So here the recipient cells will absorb the DNA fragments and convert into the donor-like structure. So this method is called as transformations. So recipient cells actively take up the DNA of the donor cells, and this nature is called as competence. The ability to uptake the DNA is called as competence. So recipient cells are always competent in nature. What are the strains of pneumococcus? There are two different states. One strain where we found a capsule is present. So the bacteria having capsule is called as capsulated, and they are very protective in nature, and they are pathogenic. And so they are pathogenic. They are virulent, pathogenic. And other bacteria where R2 strain where no capsule is present. Where no capsule is present means they are non-pathogenic. Means our WBC antibodies will kill this bacteria. So they are unable to infect. So R2 strain is non-virulent, non-disease causing. S3 strain is disease causing bacteria. So these two strains of pneumococcus are taken in the experiment. So Griffith used both the strains experimentally, and he injected one by one into the mice. So in his first experiment, he injected this into a mice, and the mice causes this because S3 bacteria causes infection. So it causes infection, so it causes death. Second, R2 bacteria, when injected, it never causes infection because so no kill of the mice takes place. But when he used heat killed S3, the heat killed S3 when injected, so no infection. No infection takes place because the capsule is being destroyed by heat. So when heat is applied, the capsule of the bacteria is destroyed, for which they are unable to infect. So when again what he did, he mixed the heat killed and R2 strands. So this one is also non-infective. This one is also non-infective. When he used both non-infective, still he find that the mouse dies. So he was a bit confused that both are non-virulent. Heat killed and R2 are non-virulent. Then why it causes death in the mouse? So experiment it is proved that the R2 bacteria contains the virulent DNA inside it, and this R2 bacteria converts into S3 strains, helps in formation of capsule and causing death. Means, for example, this is R2 bacteria. When the DNA of the S3 is incorporated, so this DNA have the information, so it forms the capsule. And as the R2 is converted to S3 by the formation of the capsule, so they becomes infective. So Griffith concluded that the transfer of certain metals takes place, and he concluded the principle is called as transforming principle, because the transforming materials from S3 to R2 causes the death of the mouse. So here what happened in the first step, S3 is injected in a healthy mouse and it causes death. RT is injected to the healthy mouse, no death, mouse suffered no infection. Heat killed S3, means the capsule is being removed out by heat killed, but the DNA is injected. When the DNA is being injected, mouse survived. But R2 living and S3 living, when both are injected, we find the healthy mouse dies. So what is the reason behind it? The reason is that this R2 is converted to S3 by competent of the DNA, and it forms a new material that is called as capsule, and this capsule causes infection in the healthy mouse and causing death. This is the main reason. So here what happened, the S3 DNA is being transported into the R2 and converting R2 into S3 form, and which causes the death. So the living mouse S that causes death. So these are the S colonies formed. This the R colonies formed. Here heat killed. When heat killed means no capsulated bacteria are found, is not collected, no colonies are isolated from the tissue because already we have killed it. But in the next stage, where R and heat killed, we find that the DNA is incorporated into the R and we find a new bacteria. Here some modified R2 are present that causing disease, and certain R are present. For example, suppose we have taken 100 R2. After transformation, we find 50S and 50 R2. So this 50S that convert into R2 convert to 52 will cause the death, will cause capsule and causes death. So this is how transformation takes place. So this experiment was again conducted by Avery, MacLeod and McCarthy. And they experimentally proved that yes, Griffith was right. In the year 1944, they introduced that yes, the DNA is the actual genetic material that helps in transformation of information and helps in formation of new capsule in the non-capsulated bacteria and causing diseases. So DNA of the dead S3 is transformed to R2, causing and converting R2 to produce the capsule. This capsule causes the death of the bacteria. So this experiment proved that DNA is the genetic material which is transferred and it gets recombined with R2. Suppose this is R2 and S3 is incorporated. So R2 and S3 are incorporated. So here this is called as transformation, or it is called as genetic recombination. Both the genes are recombined. So it is called as genetic recombination. So it is one type of sexual reproduction. Now let's move to the next method of sexual reproduction or sexual recombination, that is called as conjugations. So what do you mean by conjugation? Conjugation means union. Conjugation means union. So the conjugation was given by the scientists Lederberg and Taunton in the year 1946. So according to conjugation, they used the bacteria that is called as E. coli. So this process was experimented on E. coli bacteria. And they found that the DNA is transferred from the donor cells to the recipient cells by the process of conjugation and with the help of conjugation tube. And also the conjugation tube is formed by the help of F plasmid of F factor. Got it? So F plasmid is the fertility factor. Suppose any bacteria, suppose the bacteria having the plasmid then this bacteria will definitely produce the conjugation tube. If this is the bacteria present without plasmid, no plasmid, so no conjugation tube. No conjugation tube is formed. But in this particular bacteria, as the presence of plasmid is present, so conjugation tube is being produced. So conjugation tube is produced. So the bacteria having a plasmid or the fertility factor plasmid will produce the conjugation tube to transfer the DNA content. So the F plasmid converts F minus without plasmid, F plus having plasmid. For example, suppose this is having plasmid and this is the DNA. For example, this is only a DNA. So this is called as F plus and this is called F minus. So they will donor. So this acts as the donor. So F plus acts as the donor. F minus acts as the recipient. As F plus contains the plasmid, F plus contains the plasmid, minus don't have the plasmid. There are two different forms of bacteria on the basis of presence and absence of plasmid. So plasmid replicates during conjugation. Only plasmid present helps in formation of conjugation tube. Let's discuss. For example, suppose this is the plasmid present, and here what happens, as we have discussed that this bacteria contains the plasmid. So as F plasmid present, so conjugation tube is present. Plus here no plasmid present, no conjugation tube. So F minus, no conjugation plasmid, F plus with. So this conjugation tube will, at the time of union, this conjugation tube will extend and attach with the recipient bacteria, and they will help in dissolving the common layer of recipient bacteria by enzymic action. So a common passage is formed, and this common passage, the plasmid, one of the plasmid replicated, and the replicated plasmid moves in the recipient cells, and this recipient cell is getting the plasmid, becomes F plus, and this becomes also F plus. Initially it was F plus and it was F minus. And let's move to the next type of plasmid, that is called as HFR cells. What are HFR cells? For example, suppose this is the bacteria present and this is the plasmid present, and this is the bacteria present without plasmid. So here we call them as HF, F plus or F minus. But here what happens, these plasmid gets accumulated or integrated with the DNA. So when it gets integrated with the DNA, suppose the DNA having the plasmid is called as episome. Got it? And this episome will induce this bacteria to produce two type of DNA, and again return back to the plasmid. So this is the duplication of DNA. This is one DNA, this is one DNA. Got it? Next what happened? As the plasmid is present, they will suddenly produce the conjugation tube due to presence of plasmid, and two DNA present. They gets attached with the recipient cells, that is F minus cells. So here what happened? In the next stage, as usual, one of the DNA will transfer into it. So one of the DNA will find here plasmid, only one DNA present, here both the DNA gets attached mixed. So this is the modified form of DNA present in F minus. As episomes is getting attached to this particular cell, so these are called as high frequency FR, high frequency recombinations. So here high recombination frequency takes place that induce the DNA to duplicate into two forms, and one of the DNA gets transferred into the F minus, and converting this F minus is as is converted into a modified form. So this is how in HFR plasmid integration takes place. So here such cells are called as HFR cells, high frequency recombination cells. So high frequency recombination cells, high recombination frequency. So high frequency means is a factor present. They will convert this DNA, they duplicate the DNA, and the duplicate moves to the other DNA. This is the conjugation process. So in the conjugation process, the DNA transfers occurs through a conjugation tube. Yes, I have discussed the occurs through the conjugation tube. And efficiency depends on episome. If episome is present, maximum episome takes place, maximum conjugation takes place. And gene transfer depends upon the contact time. If the contact time between two bacteria, suppose this is the bacteria, and the contact time is more, then maximum conjugation, the maximum transfer takes place. If the contact time between the two bacteria F plus and F minus, or HFR and F minus is less, then less conjugation process will take place. So here what happened, let's go into the conjugation process just we have discussed. That is, for example, this is the plasmid present, this is the DNA present, and here this is they don't have plasmid, so F minus, In the replication, what happens? The plasmid replicates into two. One of the replication plasmid moves into the daughter cells, and this F plus initially present, now this F minus is converted. Initially F minus converted F plus. So this is how transfer of plasmid takes place. So your plasmid takes place and convert F minus to F plus. But in HFR, what happens? Then in HFR, the plasmid is present. The two DNA are present with the help of plasmid, and one of the DNA is incorporated into the daughter cells. And in the daughter F minus, we'll find two DNA present. So this F minus is a high recombinant virus, a bacteria is a high recombinant virus bacteria present. Okay. Now discuss about the transduction. So what is transduction? Transduction is the transfer of viral gene to bacteria and then to other bacteria and then to other bacteria, to bacteria and then to other bacteria. Got it? Means bacteriophage. Viral DNA genes means these are the bacteriophage. This is the bacteriophage present. Got it? So let's move. So transduction transport DNA using bacteriophage. What is bacteriophage? Virus infecting bacteria. And it was given by Zinder and Lederberg in the year 1952. And it is experimented on Salmonella typhimurium causing the disease typhoid. E. coli was experimented on conjugation, and Diplococcus pneumococcus bacteria helps for transformation experiment, and the Salmonella typhimurium is used for transduction experiment. So in transduction experiment, normally what happens? The bacteriophage present. It's the bacteriophage present. This is the bacteriophage present. This bacteriophage and is the viral DNA, bacterial DNA present. So this bacteriophage dissolves the wall of bacteria, releases its viral DNA. This phage DNA moves, and inside the phage DNA, what happens? The phage DNA either it replicates by taking the DNA polymerase enzyme of this host cell, it replicates, and this replication then breaks the DNA of the host, and this DNA is broken down and is integrated with the viral DNA, and this viral DNA again forms this capsid layer and tail region. And here your new capsid is formed, new bacteria, viral bacteriophage is formed, and they dissolves the layer of the host cell, and so they causes lysis. Breaking down of bacteria is called as lysis, and the new bacteria again results and gets attached to the new fresh bacteria. And in some case, and this phase is called as lytic phase, where lysis takes place. What is lysis? Now dissolving the host bacteria, killing the host bacteria, releasing the new bacteriophage, a new modified bacteriophage is called as lytic phase. But when we move on to the lysogenic phase, here what happens? Normally, this phage DNA, they integrated with the parent DNA, the host DNA, they gets integrated with the host DNA. After integration, they helps in replication of this host DNA. And after replication of this DNA, they then causes division and forming this bacteria. And this bacteria is of one type, this bacteria is of one type. Here, this phase is a lysogenic phase where the host is not killed. The host cell is not killed, but it is multiplied. So in lysogenic stage, we will find the host cell, the bacterial cell is not destroyed or not killed. In lytic phase, we will find the bacterial cell is destroyed and only the bacteriophage comes out. Here what happens? Now here the bacteria comes with the integrated form of DNA and called as lysogenic phase. So lysogenic bacteria, viral DNA integrates in the bacterial DNA, and such bacteria are called as lysogenic bacteria. Yes, and integration takes place. But in lytic cycle, what happens? The phage participants multiply inside the host cell and kills the host cells and release new phages to the outside. So in the lytic phase, the host cell is destroyed or killed. But in faulty phase, sometimes there is a faulty phase present where some phages carry bacterial DNA fragments, and these phages, for example, suppose this is the phage present, the viral DNA present and some bacterial DNA present, and they move to the other bacteria and release the bacterial DNA to the other bacteria, and this bacterial DNA integrates, and this is called as faulty phase of DNA. This in nature is called as faulty phases of the phases of DNA. So faulty phases infect another bacteria, yes, and release the bacterial DNA enters into it and causing a new. So all this experiment we find what? Transfer of genes takes place or transfer of DNA takes place. So in this sexual recombination is nothing but it is a transfer of genes from one bacteria to other bacteria. Yes. Then we move to types of transduction. What are the types of transduction? Generalized transductions means transfer any bacterial DNA. So in this form, any bacterial DNA can be transferred. Example, T4 phase. Specialized transformation, only specific DNA region of the bacteria is being transferred. So specific region of the bacteria is transferred, specific region. And here any DNA phase is being transferred, difference. And lambda phase carry specific bacterial genes. They never carry all genes. Specific bacterial genes are carried. Abortive, where no DNA is being carried out. If no DNA is integrated or carried to any bacterial genome, is called as abortive transduction. So no transduction takes place in abortive. So lambda phase is present, one kind. Abortive phase is present, another kind. And also we find specialized transduction and generalized transduction. Specialized transduction, where only all types of DNA can be integrated, T4 phase. And generalized transduction, in special transduction, only special DNA region is being integrated, whereas in lambda, specific bacterial genes are integrated. Abortive, no DNA is integrated into bacterial cells. So this is all about the types of transductions. Now let's discuss about archaebacteria, the primitive form of bacteria. So what are archaebacteria? So archaebacteria are the primitive or ancestral form of bacteria. So before eubacteria, this archaebacteria present, which are found in harsh habitat. So these survive in adverse condition in harsh habitat. Why? These bacteria can survive in saline water, in marshy areas, in hot spring areas, in acidic nature areas. Some lives in marshy areas, yes, as I called, northern salty marshes, also in marshy areas. Let's move into the features of archaebacteria. In archaebacteria, the cell wall is present like bacteria, but they don't contain no peptidoglycan. No peptidoglycan. They don't have peptidoglycan, that is called as murine. They don't have peptidoglycan, means peptide and glucose. So they don't have peptides and that is polysaccharides. So glycans are polysaccharides. So peptidoglycans are absent in their wall. First difference. Second difference is that they have branched chain lipids present. So normally the lipid chains are single chains, but in the membrane they have branched chain lipids present, and this branched chain lipid helps them to survive in adverse or harsh conditions. Then these archaebacteria don't have introns. Sorry, they have introns. What are introns? So in normal eukaryotic bacteria, we find exons and introns present. Exons and introns present. So exons are functional genes, and introns are non-functional genes. Mostly prokaryotic cells are exonic genes. So all prokaryotic or normal bacteria are exonic genes because they contain exons. They don't have introns, but archaebacteria do contains introns. What are introns? These are non-functional genes. So they contains non-functional genes in their chromosomes or in the DNA content. So what are the different types of archaebacteria? One is called as methanogens, methane producing bacteria. Now halophiles, which survive in salty area, saline water, thermo, high temperature, acidophiles, high, low pH, acidic condition. So these are three groups of archaebacteria. Let's start with methanogens. What are methanogens? These are special bacteria that produce methane gas from formic acid, carbon dioxide. So these are the bacteria that produces methane gas from specific formic acid or in carbon dioxide. And they live in marshy habitats. They are found in marshy areas, marshy habitats. And they are also, another very important question is, these are anaerobic bacteria. All methanogens are anaerobic bacteria. And these bacteria are found in the gut of the ruminant stomach. What are ruminant stomach? These are the compound stomach present. These are the compound stomach present. So we find compound stomach, that rumen present, reticulum present, abomasum present, omasum present. So these are compound stomach. So this is mostly present in the cow, buffalo. We find this type of stomach. So in the gut, these bacteria are found to digest the cellulose. It helps in digesting the cellulose of the plant and helps in easy digestion for the animals. And they produce, in return, they produce methane gases. So they produce methane gases, and these methane gases are used as fuel or biogas. These are called as biogas because they liberate energy. They gives fuel. So sometimes it is called as biogas. They use biogas. The best example is Coccus and Methanobacterium. The next group is called halophiles. Halophiles are the group of bacteria that survive in saline water or they are found in salty areas. And these bacteria in the membrane, in the plasma membrane, they contain a purple membrane that contains the chemical that is bacteriorhodopsin. This rhodopsin is mostly present in the retina of rods. In the eye retina of rods, we find this compound is present, but this is now found to be present in the membrane of this particular bacteria. And this membrane normally absorbs sunlight and convert the sunlight into ATP. Got it? So these purple membrane, they absorb sunlight and converts the energy into ATP. But these ATP are not used in food productions. Got it? So as they are unable to prepare their own food, so they are heterotrophic in their nature. And the best example is called as Halobacterium and Halococcus. These are the two forms of bacteria. Thermoacidophils means they can survive at high temperature and low pH means acidic condition. They can survive in acidic condition. High temperature above 80 degrees centigrade they can survive also easily. So these thermoacidophils found in hot acidic environment. And they survive at high temperature and low pH means they survive at acidic condition. So hot spring, boiling water above 80 degrees centigrade. They can survive above 80 degrees centigrade of boiling water. They also can survive above 100 degrees centigrade also. Normally these bacteria, they oxidize the sulfur to acidic form, sulfuric acid, for which they produce acidic. They're acidic in nature. And the sulfur oxidation do helps in releasing energy. And this energy is used in the preparation of food. This energy is used in the preparation of food. So normally these thermoacidophiles can prepare their own food, means they're autotrophic. What kind of autotrophic? Chemoautotropic in nature. Yes, they are chemoautotropic in nature because the energy produced during oxidation of sulfur is used in production of food. So the chemosynthetic bacteria. Example, thermoplasma and thermoproteus are the best examples. So this is all about archaebacteria or the primitive form of bacteria or the ancestor form of bacteria. One is methanogens, another one is halophiles, and the last one is thermoacidophiles. Okay, now let's discuss about eubacteria. So eubacteria are the advanced form of bacteria. Initially we have discussed about archaebacteria, but eubacteria are the true bacteria. These are the true bacteria. The best example is Anabai and Nostoc. Let's discuss one by one. So they possess a rigid cell wall, a definite cell wall, and the cell wall is made up of peptido, that is glycan. The cell wall is made up of peptides, bonds, and glucose or glycans, that is polysaccharides. So it contains rigid cell wall. Many have, not all, many of them have flagella in the cell or in its structure. Let's move into the types of eubacteria. Cyanobacteria is one subgroup of eubacteria. Then followed by mycoplasma is another subgroup. We also find actinomycete is also present, and rickettsia also present. Let's discuss one by one, that is cyanobacteria. It is sometimes called as blue-green algae. These cyanobacteria are gram-negative photosynthetic bacteria, and is the first bacteria to liberate oxygen. During photosynthesis means they contains both pH1 and also they contains pH2. So photolysis of water is found to present in this particular bacteria. So these are called as oxygenic photosynthetic bacteria because they liberate oxygen to the atmosphere. Initially, at the time of origin of life, the atmosphere was reducing atmosphere, but due to the evolution of this particular bacteria, the atmosphere regained the oxygen, so it is called as oxygenic atmosphere, and oxygen returns to the atmosphere through this bacteria. So cyanobacteria is sometimes called as the blue-green algae. The best example is nostoc and abina. They are classified under cyanophyceae, or you can classify it under mixophyceae. Now, occurrence of cyanobacteria, the habitat of cyanobacteria, they are mostly freshwater, that is aquatic. Some are found to be present in seawater, marine. Some are found to be present in moist soil. And the cyanobacteria also found to be present in high temperature water or thermal water. And they also found mutual benefit or mutual association with other groups of plants like algae, like fungi, Bryophyta, Pteridophyta or gymnosperm. In cyanobacteria, the symbiotic association is seen, that is anabina with azolla, or we find this cyanobacteria associates with a cycas root called to form coralloid roots, and sometimes fungi, algae are associated to produce lichen, and also we find this cyanobacteria present in some protista called as cyanelles. If cyanobacteria present as symbiotic association with protista, these are called as cyanelles. We do call them as cyanelle to this form of bacteria. Okay, so now move to the structure of the cyanobacteria. Mostly they are unicellular, single cell organisms. Some are present in colonial form. Some groups are present in colonial forms. Some are present in filamentous form. So the filamentous form is known as trichomes. The filamentous form is called as trichomes. So trichomes is the filamentous form. So let's discuss. This is the trichome present, and we find a mucilaginous sheath is found to be present. This mucilaginous sheath is present. Outside, this is the mucilaginous sheath present, and these individuals are prokaryotic cells. These are prokaryotic cells are found to be present, and this is called as trichomes, and it is a filamentous form, example nostoc. And trichomes are surrounded by a sheath that is called as mucilaginous sheath. They lack flagella, they don't have flagella in the cell structure. So flagella also absent throughout the life cycle. So in the life cycle, they don't find any flagella present, and these are mostly prokaryotic cells, as the nuclear membrane is absent and membrane bond organelles are also absent. And also chromatin submatrix are found to be present at the center. These are naked DNA present at the center, and the cells are surrounded by a mucilaginous sheath for protection. Outside covering present, a sheath is present outside the cell wall that provide protection to this particular wall organism, and peptidoglycan is present. This is the true cell wall composition present, or it is called as murine. So murines are found in the cell wall, that is peptide bonds are peptide and glucans are present, glycans are present, and their resemblance with the bacterial chromosome, as the chromosome is nucleate cause of the inner mass. The inner part, this one is called as the nucleoid. The circular chromosome or DNA is present, and we find the lamellosomes are present. These are the inclusion. In bacteria, normal bacteria, we found mesosomes present, but in eubacteria, BGO, we find lamellosomes present, which helps in attachment with this DNA during the time of binary fission. It helps in replications of DNA or formation of two daughter cells. And this is the mucilaginous sheath present, and this one is the cell wall present, and this is the cell membrane present, and we find these are the thylakoid membranes present with pigments present, and some cell inclusion present, like oil droplets present, gas vacuoles present, volutins present, etc., etc. So the protoplasm is divided into centroplasm and chromatoplasm. So it is divided into centroplasm and chromatoplasm. Means the outside part, the peripheral part, this peripheral part of is called as chromoplasm. Chromoplasm. Why it is called as chromoplasm? Because it is colored. Why it looks colored? Because the thylakoid membranes are present having pigments. But the inner mass is called as centroplasm. It is colorless. Centroplasm because the nuclear DNA is present. The nuclear DNA present. So this is what we find. The protoplasm contains, that is centroplasm, and chromatoplasm, which is colored form, and this is colorless form. This is colorless form. Again, we find the centroplasm contains the nuclear materials and the chromatoplasm contains the photosynthetic materials, that is thylakoid, which are colored. Cell organelles, the membrane-bound organelles are absent. Yes, they don't have mitochondria, they don't have chloroplast, they don't have, that is endoplasmic reticulum or Golgi body, etc., etc. They don't have all these things. But 70 is ribosome present because ribosome is the only cell organelle present in both prokaryotic cells and eukaryotic cells. They contains lamellar instead of mesosomes, because in normal bacteria mesosomes are present, but here we find cell inclusion, cell invagination or enfoldings of cells called as lamellazome. And this lamellazome gets attached with the nuclear membrane and helps in replication and helps in binary fission, formation of two new daughter individuals. Gas vacuoles are present that helps in floating to maintain buoyancy, which helps in floating in water. Photosynthetic pigments present in thylakoids are present in two different forms. One is chlorophyll A, another one is phycobilins. So phyco means these are the algae, bilins are the bile-like pigments present. Not the bile pigments, but the chemical composition is equivalent to the bile pigments of liver, so we do call them as bilins. Phyco means present in the algae. And the phycobilins are in the two form. One is phycocyanin, phycoerythrin. Phycocyanin produce blue color and phycoerythrin produce that is called as red color. So they contains blue color, they form body color. So phycobilins provide the body coloration, the body color, but chlorophyll A helps in photosynthesis. They helps in photosynthesis. Got it? So chlorophyll helps in photosynthesis. Phycobilins provide body colorations. The reserve food materials are present in different forms. Cyanophysin granules, reserve proteins. Then beta granules, the reserve fat droplets. Then cyanophysin starch, the reserve starch as the reserve food materials. The starch as the reserve food materials. Volutin bodies are present to resolve the phosphate. Polyhedral bodies are present to resolve the rubisco. And the reserve food matters, those presence helps in metabolism, that is, they do helps in synthesis, that is called as anabolism of the cell, and also they helps in catabolism of the cells, that is breakdown. So they helps anabolism and catabolism. And let's go to the Guy-Dukovas principles. So this cyanobacteria follow Guy-Dukovas principles, and Guy-Dukovas principles helps chromatic adaptations, mean colored adaptation. For example, suppose this is the water body present. If cyanobacteria present at the surface, it will imparts a different color. If present at the bottom of the benthic region, it will imparts a different color. So the surface, the wavelength of light imparts blue color, and in the deep, the wavelength of light imparts red color. So these two variation of color on the surface of water and the different wavelength of light absorption is called as Guy-Dukovas principle. So pigment coloration changes according to the change in the wavelength of light. So trichodesmium erythritum present in the benthic region show red color, whereas blue-green algae present on the surface show blue color. So this is about the chromatic adaptation. And let's briefly discuss about what the structure do contains. That is, they contains the gas vacuoles are present, circular chromosomes are present, that is circular DNA present, mucilaginous coverings are present, surrounding cells are present to provide protections. And also in the metabolism we find that they are self-dependent organisms, but they are autotrophic. They are autotrophic. That means they produce photosynthesis. They use carbon dioxide to release food materials and liberate oxygen by means of photosynthesis. And also they liberate nitrogen, means they are nitrogen fixing bacteria. They fix nitrogen, converts to ammonia and increase the fertility of the soil. And this nitrogen fixation in cyanobacteria is done by a specialized cell called as heterocyst. So nitrogen fixation is an anaerobic process, and it is done by a special bacteria cell called as heterocyst. So these heterocysts are present in the bacteria. These are the heterocysts present, and these heterocysts helps in nitrogen fixation because they contains the enzyme nitrogenase enzyme, and this nitrogenase enzyme only active in absence of oxygen. It is only active in the absence of oxygen. So as it is active only in the absence of oxygen, so it activates the nitrogenase enzyme and helps fixation of nitrogen. So the heterocysts are specialized cells where the nitrogen takes place, but nitrogen fixation requires the enzyme nitrogenase, and the nitrogen only works in the absence of oxygen, for which the heterocyst cell wall is very thick in nature. So it is an anaerobic nature for which the heterocyst nostoc, that is exonostoc, the cell wall is very thick. Why? They prevent the entry of oxygen and activates the enzyme nitrogenase for nitrogen fixation. So heterocysts have terminal pores. The matured heterocyst, when the heterocyst becomes matured, they have pores opening to liberate nitrogen. And this matured opening also contains certain granules present, and these granules are called as polar granules are found to be present. That do helps in fragmentations and do helps in formation of new bodies, new asexual bodies for reproduction, that is called as hormogonia. So photosystem 2 is absent in heterocyst. This photosystem 2 helps in photolysis of water. Photolysis of water, a breakdown of water to release oxygen. So heterocyst never release oxygen because photosystem 2 pigment is absent. System is absent. So oxygen is never liberated by heterocyst. But in trichomes contains photosystem 1. So photosystem 1 is active in heterocyst. But photosystem 2 is active, it is active in trichomes. So it is active in trichomes, but 1 is active in heterocyst, but 2 is active in trichomes. So PS1 generates ATP for nitrogen fixation. So this nitrogen fixation needs ATP, and to this ATP, this is used to fix nitrogen, and this nitrogen helps in increasing the soil fertility. Heterocyst also helps fragmentation. Suppose this is heterocyst. When the heterocyst matures, it comes out from the fragments. When it comes out from the trichomes, then each becomes fragments. Each are new fragments, and that develops into new individuals. And this heterocyst itself forms hormogonia. And these hormogonia are asexual bodies to give rise to new trichomes or new individuals. So nitrogen compounds release out when there is a breakage of these fragments. When the fragments breaks, the nitrogen compounds are released out, liberating nitrogen. So let's move into the reproduction. Cyanobacteria asexually. Binary fission is the most common method. Sexual method is totally absent, and multiple methods are present, like hormogonia, that is binary fission present, and we also find rhizomatic division, as we already discussed, and also we find fragmentation in filamentous form. It is unicellular form. If it is unicellular, anabinary unicellular form, they go for binary fission. If it is filamentous form, they go for fragmentation. So this fragmentation, the leak fragments develops into new individual, and these fragments gives out, that is nitrogen is liberated to them, and these fragments forms new filaments. Heterocysts germinate under special condition, that is called as hormogonia, as I have discussed, and this hormogonia forms mucilage develops around it, and these hormogonia they produce mucilage, and simultaneously what happens inside the mucilage increases in nature. Inside the nucleus, new individuals are produced. This is how we find. And some dead heterocysts, the dead trichomes cells, are called as necridia. So dead appearance of any cells is called as necridia. Akinetes. What is akinetes? In the trichomes, what happens? Certain cells accumulates reserve food materials and become thick in nature. And this reserve food materials thick in nature is called as akinetes. Under favorable condition, they comes out from the fragments, and these becomes new fragments individual. These becomes fragments, and these new akinetes then develop into germinates into new individuals. So this is how akinetes also helps in asexual reproduction. So akinetes accumulates food materials and they germinates under favorable conditions and give rise to new. What are the importance of cyanobacteria? They liberate oxygen to the atmosphere as they are oxygenic photosynthetic bacteria. There will be evolution of aerobic life. That is, initially the atmosphere was reducing. Later, by the presence of this cyanobacteria, atmosphere is ample with oxygen. So they help aerobic life to survive due to presence of oxygen. Increases cyanobacteria. Cyanobacteria and soil fertility. So it helps in increasing the nitrogen is converted into ammonium compounds, will increase the soil fertility, is increased due to the decay of this bacteria. The best example is nostoc and Tolipothrix, which are used in rice fields. Anaban is also used sometimes. Cyanobacteria shows symbiotic association in plants. Nitrogen compounds supplied to the host cells, and both partners are benefited. Spirulina is a cyanobacteria which is a single cell protein, and that contains heavy deposits of protein materials present rich in proteins, which are used as space food materials. Spacecraft use this as a protein supplements. And extracts of lignema, the external lignia helps in antibiotic formations. Microcyst aurigonosia causes algal blooms. So these are the harmful aspects. Also, Anavena floss aqua also floss booms. Booms causes toxins to the water materials. So toxins are produced which are harmful to the aquatic animals. So water sources become very harmful to these animals. Oxygen in the water goes on decreasing. Due to the presence of algal bloom, the oxygen decreases in the water. Aquatic animals may die. Aquatic animals may die. So this is all about, that is mycoplasma. This is about cyanobacteria. So we have completed cyanobacteria structure. It's a life process. Its uses is a disuses. Now let's start mycoplasma. So what is mycoplasma? Let's discuss about mycoplasma, its structure life process. So the scientists Snooker and Rocks first discovered mycoplasma, and this mycoplasma are discovered from the isolated pleural fluid of any organism. It is present in some other organisms, and the fluid isolated from that particular organism contains this mycoplasma. And this mycoplasma was initially regarded as pleuropneumonia-like organism. It is called as pleuropneumonia-like organism, or it is sometimes called as jokers of the plant kingdom. Pleuro means it's changed its structure. As it's changed its structure, it is called as pleuro, and it look like pneumonia-like structure, or it is called as the jokers of the plant kingdom. Mycoplasma, mycoides are classified under mycoplasma group, and this mycoplasma belongs to the class that is called as Mollicuta, for which this Mollicuta is called as Mollicutes amelos. What are amelos? Mollicutes like organisms. And this mycoplasma infect both plants and animals. The host of mycoplasma are both plants and animals. Like in plants, it infects the potato plant, corn plant, brinjal plant, etc. etc. In animals also, it causes a urine infection, mycoplasma ureate, an organism that causes urine infection in animals, in humans also. Now let's move to the structure of mycoplasma. So mycoplasma, unicellular organism, single cell structure, and the cell wall is absent. They don't have any cell wall, so they present cell membrane, which is trilamellar. But our cell membrane is bilamellar. But mycoplasma cell membrane is trilamellar, means triple layer present. So it is called as trilamellar cell membrane. They are highly pleomorphic, means they change its shape. They can change its shape. So these are highly pleomorphic because they goes on changing its shape. Shapes are spherical or filamentous, means they are pleomorphic. They can change its shape simultaneously. The cell membrane is made up of lipoprotein layers, but it is trilamellar, 3-layer. Cytoplasm contains ribosome because they are prokaryotes. As they are prokaryotes, mycoplasma prokaryotes, they contains ribosomes, but they don't have membrane bound organelles, and also they don't have, that is called as nuclear membrane. Got it. So this is the lipoprotein layer, and this lipoprotein layer present here is a trilamellar. This is a trilamellar membrane, trilamellar. And inside we find certain metabolites or chemicals are present. Special bodies are present called as elementary bodies, and soluble proteins are also present. The DNA present, the chromatin metal is present throughout the cell. It is not restricted at the center. It is present throughout the cell. And soluble RNAs are present, enzymes are present, 70S ribosomes are present, and inside the DNA we find special structures present, that is called as the disc-like structures are present. And these disc-like structures are called as replicating disc that do helps in replications. So this is the basic structure of mycoplasma. So the cytoplasm, we find nucleus, which is organelle nucleus is absent, means no cell nuclear membrane, no nuclear membrane. They don't have nuclear membrane at all. No membrane bound organelles present. That is, membrane bound organelles like mitochondria, plastids are absent. DNA and RNA are present. Yes, they are present. But mycoplasma is called as the fried egg apparatus. So the mycoplasma, it is pleomorphic in nature. So it looks like the fried egg. How the fried, when we fried an egg, how the egg looks, it looks like that. It contains the opaque middle structure and a transparent outer structure. So it's a fried egg apparatus. It looks like a fried egg apparatus. The DNA is single-stranded and double-stranded. Both DNA is found. Replicating discs are present that helps in replication, as I have told. Cytoplasm also contains enzymes for metabolic activities. Now, mycoplasma can pass through bacterial filters. So they are very minute than bacteria. So these are regarded as smallest cell. So they lack cell wall, and sterols are required for its growth. For growth, it needs sterols for the growth. And the nutrition is heterotrophic. Mostly they are unable to produce the food, so they are heterotrophic in nature. Some are saprophytic, means they depend on dead organic matter. And most are parasitic, means they are pathogenic, causing diseases in plants and animals. As they are pathogenic, they live in living host. And the smallest prokaryotic cells, called as mycoplasma gallisepticum, is regarded as smallest cell, that is PPLO. And the smallest cell range between 0.3 to 0.1 micrometer. And mycoplasma survive without oxygen, so they can go for an aerobic condition. These anaerobic mycoplasma, they are very sensitive to antibiotics, like mycoplasma sensitive to penicillin. They are highly sensitive to penicillin because mycoplasma don't have cell wall. So penicillin can easily enter into the mycoplasma and act on it. They are also sensitive to streptomycin because mycoplasma don't have cell wall. They are sensitive to chlorophenicol also. Antibiotic resistance. Mycoplasma are resistant to erythromycin because these erythromycin, they can enter in the erythromycin because mycoplasma don't have the cell wall due to absence of peptidoglycan. So all these antibiotic resistant genes can enter into it. Cell wall synthesis inhibitors are ineffective. Reproduction in mycoplasma. Normally the reproduction in mycoplasma is not so known as it is prokaryotic cell. So the reproduction is mostly by binary fission. Not clear known, but elementary bodies may be present in it, and this elementary body helps in division of this bacteria. Mycoplasma is very simple. Bacterial structure is very simple. This mycoplasma bacterial structure. Now move into the different facts of this chapter. We have completed the chapter Monera. Now let's discuss about different facts of this chapter. Means cyclosis is absent in bacteria. Means the streaming movement of cytoplasm is absent in bacteria. Flagella consists of protein called flagellin protein. Bacillus is the most common form of bacteria. And Pseudomonas putida is called a superbug. Pseudomonas putida is called as the superbug. Methanogens are called as obligate anaerobes. These are anaerobes, methanogens. Thermoacidophils are facultative anaerobes. Obligate anaerobes are methanogens. Facultative anaerobes are thermoacidophiles. Archaebacteria wall contains, that is, N-acetyloaminomuronic acid, and exogenotransfer occurs from HFR cells. Mycoplasma are facultative anaerobes. Thermoacidophils are facultative anaerobes. Mycoplasma are facultative anaerobes, whereas that is normal bacteria, they are of aerobes, obligate. Heterocyst and Vibrio are facultative anaerobes. These are very important examples in questions. Nitrogen occurs in heterocyst. Bacteria show extensive metabolic diversity. So bacteria has extensive metabolic diversity. So the metabolic diversity in bacteria is very extensive. Now let's discuss about some practice questions. So you pause this video and go through this question. Select the incorrect statement with respect to eubacteria. Eubacteria have very simple structure. Yes. Petroglycan nature cell wall is present. Yes. Heterotrophs are most abundant in nature. Yes. So most simple metabolic diversity. So this is D. You go, as D is right or wrong. Which one is wrong? Find the odd one with respect to phototropic nutrition. Chromatium and chlorobium. Rhodopseudomonas and trichospirillum. Which one is phototropic nutrition with respect to phototropic nutrition? As we know, this chromatium, chlorobium, all this we have discussed. The chloronema and chlorophyllus, pseudomonas and clostridium. Okay, the third question, mark the incorrect option with respect to nitrogen fixing bacteria. Nitrococcus, leptothrix, yes. Nitrobacter, yes. Nitrocystis. Which one? Then the fourth, genetic recombination in which a small doubles in DNA is transferred in the donor bacterium by bacteriophage was first demonstrated by the fellow Griffith or Lederborg and Totem or Ginder and Lederborg or Avery and et al. Go through it and just give the answer to my WhatsApp or give the answer to our channel present, and let's see how many of you are right, how many of you give correct answer. Select the correct match. Column 1 we have rating of fibers or removing the bast fibers from the jute. Pollution control, as we have discussed in this chapter, you go through it and answer all these questions. Curing of leaves and bacitration. Column 2 contains Pseudomonas putidia, as we have discussed now. Pseudomonas fluorescens, Bacillus licheniforms, Micrococcus canadians, and these are the options. Go through the options. Then move on. This is archaebacteria do not show so and so peptidoglycan in cell wall, introns and DNA. Branch lipid cells they have. Ribosomal proteins are highly acidic in nature. They have. Thermoacidophils are capable of withstanding extreme low pH, yes, and high temperature. Yes. Why? Not due to presence of branch chain lipid in cell membranes. Presence of resistant enzymes, yes, which can operate in basic conditions. Presence of higher concentration of KCl in the cell, or more than one option is correct. Find out. Which group of Monerans played significant role in the evolution of aerobic forms of life? Monerans. That is mycoplasma, cyanobacteria, you know very well, just I have discussed, archaebacteria or actinomycetes. The characteristic photosynthetic pigments in cyanobacteria are chlorophyll A and C, chlorophyll A and carotenoids, chlorophyll A and phycobilins. Chlorophyll A, carotenoids and phycobilins. Go through it and give the right answer. And the smallest wall-less monerans means they don't have wall, means the smallest prokaryotic cells, as we all know. You can go through it. Develop fry egg appearance in culture, yes. Are motile, have definite shape, are mostly saprophytic. You go through the questions. And to practice 1000 above questions or more than 700 plus questions with online and normal practice questions, you can go through this channel. Or online exam, you can give online exam in this particular site, or you can go for doubt clearing classes in this particular site. You can get free PDF, mind maps, worksheets, MCQs in this particular site. You can contact this number and you can get the study materials also from our Telegram channels. Hope you all enjoyed the video or the class, and thank you.